Hello, students. Welcome to KENE'S Educare, a group of KENE solutions. This is part 3 of lesson number 5, the fundamental unit of life. Okay, so in this lesson, we have seen like how our human body has different, you can say, organs which have been situated for performing the certain functions of our body. The same way, the cell that our body is made up of, we are all aware of that our human body is made up of cell. We are in multicellular organisms, right? हम क्या है? Multicellular organism है. And the and you know this thing that when a group of cell come together, that makes up a tissue. When a group of tissue come together, that makes up an organ. When a group of organ come together to perform some vital functions of a body, then we see that a multicellular organism is functioning or it is performing its function, right? So this is how our human body is been formed and how are we having these organs and all. So last class may we have discussed on the first, you can say, first component of the cell that we are having. Before that we have discussed on uh, cell where we have seen that Robert Hooke sir, he was the first one जिन्होंने सबसे पहले सेल को डिस्कवर किया था in the year 1665, right? And we all know that cell is the structural and functional unit of life because cell जो है उससे ही तो हमारी बॉडी बनी है. The smallest thing with which our body is made up of is the cell, but it has certain functions to perform, and for performing that function, okay, वो function perform करने के लिए it has certain components. जैसे कि हमारे body में we have our heart that is performing the function of pumping the blood, right? Then we have our kidney that is helping us in filtration. We have our liver, we have our lungs that is helping us for respiration, right? So वैसे ही cell के अंदर भी different components हैं which are helping the cell to carry out some vital functions of our body. Okay, and those functions that the cells are performing, those are an important function. जैसे कि last class में we have studied about the plasma membrane, which is also called as the cell membrane, where we have discussed on diffusion and osmosis concept, right? हमने diffusion and osmosis पे discuss किया था last class में. There we saw that how the plasma membrane, it is selectively permeable, and how the diffusion and osmosis process goes on from a higher concentration level to a lower concentration level, right? So we have discussed that. Now we are going to discuss on two different activities which have been given. When we start, we are going to discuss on two different activity, and then we have the second component of our cell, that is the cell wall. Cell wall के बाद we have certain different components as well which have been given here, right? So see, we started by studying about the cell theory which was given by Robert Hooke sir. Okay, so he had given the cell theory in the year 1665, and just imagine the condition when there was no microscope, there was no research, there was nothing at all to come up with this minute concept of cell. Then too, this Robert Hooke sir, he had made this stance where he has observed this cell ka structure through a self-made microscope or a self-designed microscope. So he was relating this cell ka structure somewhat to the structure of the honeycomb as well as the structure of the cock, because he was finding it that the structure of the cock is resembling the structure of the honeycomb, because as we all know that honeycomb के अंदर small small compartments है. So he was telling that cells also look the same, like the small small compartments they are present. Okay, so he Robert Hooke, he had called those as boxes cell. क्या बोला था उन्होंने? Boxes cell. Okay, the little compartment that he was observing, he told that as boxes cell. Okay, so now he told us like he was the first one who has discovered the cell. So it was discovered by Robert Hooke in the year 1665, and cell जो है, that is a Latin word which means a little room. Cell का मतलब क्या है? Little room. Okay, so cells, they represent the basic structural and functional unit of life. क्या represent करते हैं cell? Structural and functional unit of life. And we have certain organelles. Now organelles are small structures which are present inside the cell, and they, like these small structures, they are going to perform some specific functions of the cell. Okay, so all the organisms that we have, they are composed of one or more cells. All the organisms that we can see around, they are made up of one or more cells. And all the tissues and the organs that we have, they are indirectly composed of cell, because cells, when a group of cells come together, they make up a tissue. When a group of tissue come together, they make up an organ. So all the tissues and the cells, they are directly composed of cells, right? So we can conclude from this that there is no life which can exist without cell. There is no life without cell. If cell is not there, there will be no life which will exist. Okay. फिर हमने ये एक्सपेरिमेंट देखा वे वी स्टार्टेड दी सेल्स ऑफ द अनियन पील. हमने क्या किया, अनियन का पील लिया. इनसाइड वी हैव टेकन द इनर लेयर ऑफ द अनियन का पील, एंड देन वी आर गोइंग टू पुट दैट इनटू द वाटर बाथ सो दैट इट हैज़ इनफ ऑफ द वाटर. ओके, वाटर ग्लास में रखा. उसके बाद वी टुक दैट पील ऑन द स्लाइड. उसके ऊपर हम लोग 1 ड्रॉप ऑफ सेफ्रॉन इन सोल्यूशन डालेंगे, एंड देन वी आर गोइंग टू add a cover slip. And while adding the cover slip, we need to take care that there should be no bubble formation which will take place. Okay, for that we need to use a mounting needle. तो आप वो पील के ऊपर माउंटिंग निडल रखोगे, एंड देन यू आर गोइंग टू पुट दैट कवर स्लिप सो दैट बबल ना आए उस पर. उसके बाद we have this temporary amount of onion peel, that is the slide which is ready with us. So now we are going to observe that under a compound microscope, and now we have to observe it under high power lens as well as low power lens. Okay, तो लास्ट क्लास में तो भी हम लोग ने ये डिस्कस किया ही है. वी ऑल नो हाउ द structure of this onion peel looks like under the microscope, right? Even you might have drawn that. So this is how the structure of the cell of the onion cell looks like under the microscope, where you can see the structure of the cell along with the nucleus. Now here the question comes कि अगर हम अलग अलग onions लेंगे different sizes के, will the cell remain the same? ऐसा ही cell दिखेगा क्या हमें? So the answer to that we gave was yes, all the structures, they are going to look similar to each other, and together when you bring all these structures, they are going to form a big structure like an onion bulb. वो कैसा structure बनाएंगे? They are going to make a big structure like an onion bulb, and we can find out from this onion bulb wala activity that different sizes, they are going to have similar small structures which will be visible under the microscope. Okay, so the cells of the onion peel, they are all going to look the same regardless whatever size they are, whatever size of onion we are having, or from whatever size of onion they come from. Okay, so this small structures that we see, okay, ये जो आपको सेल का स्ट्रक्चर दिख रहा है, सेल दिख रहा है अनियन का, that is the basic unit of the onion bulb. क्या है वो? That is the basic unit of the onion bulb. Okay, so we saw that the structures that we can see under the microscope, we call them as cells. And not only onions, but whatever kind of organism that we observe around us, they are all made up of cell. किससे बने हैं वो? Cell से बने हैं. However, there are also single cells that live on their own. तो ऐसा नहीं है that cells are always present in number. The cells can be present as one, like there can be a single cell as well which can exist, and they are going to live on their own. They are going to have an independent life which they are going to live there. Okay, then we like this, the picture that you can see on the screen, the figure 5.3, these are the shape you can see of different cells which are present in the human body. So in human body we have this smooth muscle cell, we have blood cells, we have nerve cells, we have fat cells, bone cells, ovum and sperm. These are the various cells which are found in our human body. Okay, so the shape and size of cell, they are all related to specific function that they perform. Now whatever kind of shape and whatever kind of size that you can see of the cell, as you all can see that all the cells, they are having different shape and different sizes. Why is it so? So the shape and the size, they are all related to the specific function they have to perform. So we have some cells like amoeba. अब अमीबा जो है वो अपना शेप हमेशा चेंज करता रहता है. ओके, सो इन सम केसेस यू विल सी that the cell shape, it could be more or less, it can be fixed or it can be peculiar, that is it can be different for a particular type of cell as well. For example, nerve cells, they have a typical shape. Nerve cells kya hai? Nerve cells can have a typical shape. So each living cell has the capacity to perform certain function that are the characteristics of all form. Now हर एक living cell के पास एक capacity होती है that they are performing some basic functions. And when you see each living cell is having this capacity, it means that is going to be the characteristic which is being performed by the all living form, right? So here the question comes is, how does a living cell perform this basic function? एक living cell ये basic functions कैसे perform करता है? So here we came to know that कि multicellular organisms के अंदर we have division of labor which occurs. क्या होता है? We human beings are multicellular organisms, right? So we have this division of labor which occurs, and division of labor means what? We are having different parts in our human body, and this different parts in a human body is performing different functions. Okay, so likewise, as I said, that the human body has a heart to pump blood. हमारे पास heart है, and heart is helping us in pumping the blood. We have a stomach. Stomach is हमें कहाँ पे help कर रहा है? Stomach is helping us to digest the food. So this is how we are having different parts which are being present here. Okay. So see, similarly as I told you that we are having division of labor which is occurring from multicellular organisms, वैसे ही there is a division of labor which can be seen within a single cell as well. And each cell, they have got certain specific components within them, and we call them as cell organelles. Okay, so each kind of cell organelle, they are going to perform a specific function. जैसे कि they are going to help us in making some raw materials for the cell. They are going to help us in clearing the waste material from the cell, and this is how they are going to help us. Okay, so a cell is able to live as well as to perform its function because of this organelle. Now, how is the cell performing its function? How it is able to live? That is all because of the organelles that are present. Okay, so this organelle, it together constitute the basic unit which we call it as cell. Okay, तो ये भी जो सारे organelles है वो क्या है? These are the basic unit of the cell that we are having. And it is interesting that all the cells are found to have the same organelles. जितने भी cell है हमारे पास, they are going to have the same organelle. They are going to have the cell membrane, they are going to have the cell wall, they are going to have vacuole, mitochondria. These are the different organelles which are present in the same cell, and no matter what the function is performing, what organisms they are having, like what organism they are found in. Okay, so this is how they are going to perform their function. Wait. Okay, See, then we saw what is the cell made up of, and the question we can reframe it as what is the structural organization of the cell. So as I told you that cell has certain special components which are present inside them, which we call it as cell organelles, right? So when we are studying a cell under a microscope, हमें तीन फीचर्स देखने को मिलते हैं हर एक सेल के अंदर. How many features? We have three features which can be seen in every cell. So firstly, we have a plasma membrane, we have a nucleus, and we have a cytoplasm which can be seen inside a cell. Okay, so all the activities inside the cell and the interaction that the cell is having with its environment, all these things are possible because of these features. Okay, now let us see how this is possible. See, firstly, we have discussed on the plasma membrane, which is also called as a cell membrane. There we saw that हमारे सेल के पास एक आउटर मोस्ट कवरिंग होती है, एंड ये जो आउटर मोस्ट कवरिंग है वो सेल के कंटेंट्स को सेपरेट करती है उसके एक्सटर्नल एनवायरनमेंट से, बिकॉज द content of the cell which is inside it should be separated with the external environment. So for that we have a plasma membrane which is also called as a cell membrane in between. Okay, now the plasma membrane, because it is selectively permeable, it is going to allow or it is going to permit the entry and exit of some materials in and out of the cell. ठीक है, तो ये कुछ materials को ही allow करेगा in and out of the cell. So for this we need to say how does the movement of this materials occur or how it is moving out of the cell. So we have substance like carbon dioxide and oxygen which is moving across the cell membrane through a process called as diffusion. What is diffusion? When some substances like carbon dioxide or oxygen, when they are moving across the cell, that process we call it as diffusion process. क्या बोलते हैं हम उसको? Diffusion. So we have already studied the process of diffusion, right? डिफ्यूजन क्या होता है? डिफ्यूजन जो है दैट इज़ अ प्रोसेस, दैट इज़ अ स्पॉन्टेनियस मूवमेंट दैट इज़ अकरिंग ऑफ अ सब्सटेंस. क्या? स्पॉन्टेनियस मूवमेंट ऑफ अ सब्सटेंस दैट इज़ टेकिंग फ्रॉम अ रीजन ऑफ हाई कॉन्सेंट्रेशन टू अ रीजन ऑफ लो कॉन्सेंट्रेशन, दैट इज़ डिफ्यूजन. तो अभी यहाँ पे डिफ्यूजन में वी हैव दिस गैशियस एक्सचेंज विच इज़ टेकिंग प्लेस दैट इज़ बिटवीन द कार्बन डाइऑक्साइड एंड द ऑक्सीजन. तो यहाँ पे क्या होगा? There will be spontaneous movement of substance which occurs from a region of high concentration to a region of low concentration. Okay, so after that we discussed on, see, something similar to this happens. For example, जैसे कि substance है हमारे पास CO2, carbon dioxide. Now we know that carbon dioxide is the cellular waste and it is being secreted out by the cell. So what has been done here? सपोज कार्बन डाइऑक्साइड जो है वो सेल के अंदर आके बहुत ज़्यादा हाई कॉन्सेंट्रेशन में एक्यूमुलेट हो गया है, जमा हो गया है. सो नाउ अगर आप सेल का एक्सटर्नल एनवायरनमेंट देखोगे तो वहाँ पे कार्बन डाइऑक्साइड का कॉन्सेंट्रेशन कम है. क्या है? कार्बन डाइऑक्साइड का कॉन्सेंट्रेशन कम है, बट वहाँ पे सेल के अंदर ज़्यादा है बट सेल के बाहर का कॉन्सेंट्रेशन कम है, राइट? सो एज सून एज वी आर सीइंग दैट देयर इज एन डिफरेंस ऑफ कॉन्सेंट्रेशन, CO2 जो इनसाइड जो है एंड आउटसाइड द सेल है दैट इज डिफरेंट. सो व्हाट हैपेंस? बिकॉज इनसाइड द सेल द CO2 का कॉन्सेंट्रेशन वाज मोर, व्हाट हैपेंस? CO2 इज गोइंग टू मूव आउटसाइड द सेल क्योंकि बाहर की तरफ CO2 का कॉन्सेंट्रेशन कम है. So this is what we have discussed in diffusion. It is going to move out from a region of higher concentration to a region of lower concentration through a process of diffusion, right? So similarly, oxygen is going to enter the cell by the process of diffusion. क्या होगा? Oxygen enter करेगा cell के अंदर by the process of diffusion when the level or concentration of CO2 inside the cell decreases. Okay, and that is why we can say that diffusion is very an important role, which is with respect to gaseous exchange between the cell as well as the cell and its external environment के लिए. Okay, so not only carbon dioxide and oxygen they are following this diffusion, water also obeys the law of diffusion. क्या water also obeys the law of diffusion, where you will see that there is a movement of water molecule that is taking place through a selectively permeable membrane. Now selectively permeable membrane is our plasma membrane. तो यहाँ पे water move करेगा एक selectively permeable membrane से, and that membrane we call it as osmosis. Okay, so the movement of water that is across, ओके, तो ये जो मूवमेंट है वाटर का अक्रॉस द प्लाज्मा मेम्ब्रेन, दैट इज गोइंग टू गेट अफेक्टेड बाय द अमाउंट ऑफ सब्सटेंस विच इज डिजॉल्व्ड इन वाटर, कि कितना अमाउंट ऑफ सब्सटेंस वहाँ पे डिजॉल्व हो रहा है, उससे हमारा प्लाज्मा मेम्ब्रेन का जो मूवमेंट है, वाटर का जो मूवमेंट है अक्रॉस द प्लाज्मा मेम्ब्रेन, दैट इज गोइंग टू गेट अफेक्टेड. ओके, सो दस वी कैन से कि ऑस्मोसिस जो है दैट इज अ पैसेज ऑफ वाटर वेयर द वाटर मूव्स फ्रॉम a region of high water concentration through the selectively permeable membrane to a region of low water concentration. and ये कब तक होगा, कब तक higher concentration से lower concentration में passage होता रहेगा, water is going to get transported till an equilibrium is reached. जब तक ये equilibrium नहीं आएगा तब तक it is not going to happen. Okay, so see, the next question that we have is, what will happen if we put an animal cell or a plant cell into a solution of sugar or salt? तो यहाँ पे हमने तीन डिफरेंट कंडीशंस पढ़े थे. एक था हाइपोटोनिक सोल्यूशन, एक था आइसोटोनिक सोल्यूशन, एक था हाइपरटोनिक सोल्यूशन. राइट, हाइपोटोनिक सोल्यूशन में क्या होता है, इफ़ द मीडियम विच इज सराउंडिंग द सेल, वहाँ पे अगर वाटर कॉन्सेंट्रेशन ज़्यादा है वो मीडियम के अंदर, सेल से ज़्यादा वाटर कॉन्सेंट्रेशन है, then the outside solution is going to be very dilute. And here, because the outside solution is dilute, the cell is going to gain water by the osmosis process, and this solution we call it as an hypotonic solution. Okay, and when there is an hypotonic solution, when the cell is gaining the water, okay, that time what happens, cell का size increase हो जाएगा, right? So the cell is likely to swell up when it is an hypotonic solution, because the water is entering inside the cell. That is the reason the cell is going to swell up. Then second solution we have is isotonic solution. यहाँ पे there will be no movement of water. Now when such conditions can occur, अगर the amount of water which is present inside the cell is exactly the same the amount of water which is present outside the cell. If this condition occurs, it means the solution we call it as an isotonic solution. Okay, and third we have is hypertonic solution. यहाँ पे क्या होगा, यहाँ पे सेल के बाहर का जो वाटर कॉन्सेंट्रेशन है वो कम होगा एंड सेल के अंदर का ज़्यादा होगा. तो एज वी नो दैट इट मूव्स फ्रॉम हायर रीजन टू लोअर रीजन, तो सेल के अंदर से नाउ द वाटर इज गोइंग टू कम आउटसाइड द सेल. सो व्हाट हैपेंस, इंस्टेड द सेल इज गोइंग टू श्रिंक डाउन, सेल का साइज छोटा हो जाएगा बिकॉज वाटर फ्रॉम इनसाइड द सेल हैज़ मूव्ड आउटसाइड. राइट, सो हियर this type of solution we call it as an hypertonic solution. Okay, so osmosis क्या है, osmosis is a special case of diffusion that is taking place through a selectively permeable membrane, that is plasma membrane, which we also call it as cell membrane. Okay, now see, this is what hypertonic में क्या होता है, सेल shrink हो जाता है because सेल के अंदर से water is going out. Can you see, H2 is moving out of the cell because सेल के अंदर का water concentration ज़्यादा था as compared to the external environment cell. Okay, so that is the reason water from inside the cell it moves outside the cell. So this is hypertonic solution. Then we have isotonic solution, जहाँ पे the water concentration inside the cell as well as the water concentration outside the cell is equal, it is similar, so that type of solution we call it as isotonic solution, because there will be no net movement of the water that you're going to see here. Okay. And thirdly, we have this hypotonic solution, jahan pe water concentration outside the cell is more, and that is why the water from outside the cell is going to move inside the cell, and that is the reason हाइपोटोनिक सोल्यूशन में वाटर जो जो हमारा सेल है वो स्वेल कर जाता है, इट स्वेल्स अप because of the more water that is entering inside the cell. Okay, so these are the three types of solution that we have: hypertonic solution, isotonic solution, and hypotonic solution. Okay, I hope you have drawn this in your notebook. Now we have two different activities to discuss. So the third activity that we are having is osmosis with an egg. So we are going to discuss an osmosis with an egg. Okay, what you need to do is remove the shell of an egg by dissolving it in dilute hydrochloric acid. हम क्या करेंगे, एग का जो शेल है उसको रिमूव करेंगे. कैसे? उसको हाइड्रोक्लोरिक एसिड में डाइल्यूट करके उसको डिसॉल्व करेंगे हम किस में? डाइल्यूट हाइड्रोक्लोरिक एसिड में. ओके, नाउ द शेल इज मोस्टली कैल्शियम कार्बोनेट. जो एग का शेल होता है वो क्या होता है? दैट इज कैल्शियम कार्बोनेट. ओके, शेल क्या होता है एग का? दैट इज कैल्शियम कार्बोनेट. सो अ थिन आउटर स्किन नाउ एनक्लोजेस द एग. अब क्या है हमारे पास? जो हमारे पास एक थिन आउटर स्किन है, दैट इज गोइंग टू हैव द एग. सो यू नीड टू पुट दिस एग इन प्योर वाटर. क्या करोगे? आपने अभी एग का शेल निकाल दिया, एंड वी नो दैट द एग शेल इज मोस्टली कैल्शियम कार्बोनेट. एंड नाउ वी आर लेफ्ट विथ, आफ्टर वी हैव रिमूव द शेल, वी आर लेफ्ट विथ अ थिन आउटर स्किन. एंड वो थिन आउटर स्किन के अंदर क्या है? वी हैव द एग. So now we are going to put this egg in pure water and we are going to observe it after 5 minutes. आपने क्या किया है? एग का शेल निकाल के एग को प्योर वाटर में डालोगे एंड देन आप उसको 5 मिनट तक ऑब्जर्व करोगे. नाउ टेल मी व्हाट डू वी ऑब्जर्व? क्या ऑब्जर्व करेंगे हम? कि जो हमारा एग है दैट इज गोइंग टू स्वेल. क्यों स्वेल कर रहा है? बिकॉज उसके अंदर वाटर गया है बाय द प्रोसेस ऑफ ऑस्मोसिस. ओके, what happened is egg जो है वो swell किया है because water is passing into it by the process of osmosis. So now we have to place now what are you going to do? You have to place a similar de-shelled egg. आपने पहले शेल को निकाल दिया था by giving this dilute hydrochloric acid का ट्रीटमेंट. तो आपने क्या देखा? that the egg जो था it has swollen up because of the water which gets added into it by the process of osmosis. अब हम सेम चीज़ करेंगे पर यहाँ पे हम de-shelled egg डालेंगे. पर ये किस में डालेंगे हम? concentrated salt solution में. आपको शेल निकालना नहीं है. You have to put the egg in concentrated egg solution and then you have to observe it after 5 minutes. So आप देखोगे यहाँ पे egg जो है वो shrink कर जाएगा, छोटा हो जाएगा. Why this happens? Because अब water जो है that moves out of the egg into the salt solution. Because salt solution और भी ज़्यादा concentrated था, and that is the reason what happens? That is the reason it is going to come out of the, like, the water concentration is going to the water is going to lose into the salt solution that we are having because the salt solution is more concentrated. Okay, so we can also try a similar activity with dried raisin or apricots. तो यहाँ पे हमने एक बार शेल को we have removed that shell by giving that treatment of the dilute hydrochloric acid. तो हमने वहाँ पे देखा that because of the osmosis process egg swell up हो गया था because of the you can say water which gets entered into it. Okay, एंड नेक्स्टली हमने क्या किया, हमने डीशेल्ड एग डाला एंड वी आर गोइंग टू ऑब्जर्व दैट फॉर 5 minutes. Okay, ओके, सो यर वी सॉ कि हमारा जो एग था वो श्रिंक हो गया बिकॉज उसके अंदर से जो वाटर था दैट वेंट डाउन. वो किस में चला गया, वो सॉल्ट सोल्यूशन में चला गया बिकॉज द सॉल्ट सोल्यूशन वॉज मोर कॉन्सेंट्रेटेड. ओके, so now the next activity, one more activity we are having, where as I told you that we are going to see that with respect to we are going to see it with respect to dried raisin and apricots. So can you see this is the watch glass with dry raisins which has been kept, and this is the swollen raisin. Okay, so you need to put dried raisins or apricots in plain water and leave them for some time. क्या करेंगे हम, जो हमारा ड्राइड रेजिन है यार, अगर हम एप्रिकॉट्स ले रहे हैं तो हम उसको एक प्लेन वाटर में रखेंगे एंड वी हैव टू कीप दैट इन द वाटर फॉर सम टाइम. ओके, देन प्लेस देम इनटू अ कॉन्सेंट्रेटेड सोल्यूशन ऑफ शुगर और सॉल्ट. अब पानी के अंदर रखने के बाद हम उसको डालेंगे एक कॉन्सेंट्रेटेड सोल्यूशन ऑफ शुगर या सॉल्ट के अंदर, एंड व्हाट आर यू गोइंग टू ऑब्जर्व इज यू विल सी दैट ईच is going to gain water and they're going to swell when you are placing that in the water. जब आप उसको water raisins को water के अंदर रखोगे, the raisins are going to swell up because water from outside enters inside the raisin, and that is the reason they're going to gain that water and they're going to swell up. Okay, however, when you place them in concentrated solution, the raisins are going to lose the water and they are going to shrink because water from inside the raisin they are going to lose out, because as we know that it moves from higher concentration to lower concentration. So when there was concentrated solution, what happens, there was no water, so water from inside the raisin is going to move out and the raisins are going to shrink. So this is the same result that we got with respect to the eggshell, right? So unicellular freshwater organisms and most plant cells, they tend to gain water during osmosis, through osmosis. तो हमारा जो यूनिसेल्युलर फ्रेश वाटर ऑर्गेनिजम्स है और जो प्लांट सेल्स है, व्हाट डू दे डू, what are we going to do, we are like all these freshwater organisms और plant cell को उनका water कहाँ से मिलता है, they gain this water through osmosis process only, and the absorption of water by the plant root. आपको पता है हम roots में पानी डालते हैं बट वहाँ पे पानी जमा होता है क्या, नहीं होता है. why, because the water is being absorbed by the roots of the plant, so that is also an example of osmosis. So thus diffusion is important when there is an exchange of gases taking place, and when there is an exchange of water taking place, diffusion is a very important concept that comes here in the life of the cell. Okay, so in addition to this, the cell also obtains its nutrition from the environment. तो इसके साथ सेल जो है वो अपना न्यूट्रिशन अपने एनवायरनमेंट से भी लेता है, एंड देयर आर डिफरेंट मॉलिक्यूल्स who move in and out of the cell through a type of transport which requires the use of energy. Now we have different molecules as well who are going to move in and move out of the cell because they have this type of transport, and this transport it requires the use of energy, so that they will have to give energy. So plasma membrane is flexible and it is made up of organic molecules called as lipids and proteins. तो हमारा plasma membrane क्या है? Our plasma membrane is flexible, and what it is made up of? It is made up of lipids and proteins. Okay. However, we can observe the structure of the plasma membrane only through an electron microscope. तो आपको अगर प्लाज्मा मेम्ब्रेन का स्ट्रक्चर देखना है तो यू कैन ऑब्जर्व दैट, बट दिस कैन बी पॉसिबल ओनली by the use of the electron microscope. Okay, so the flexibility, as I told you that the plasma membrane is flexible, so the flexibility of the cell membrane, it also enables the cell to engulf the food and other material from its external environment. तो ये जो cell membrane की flexibility है, that is going to enable the cell so that it can engulf, so that it can take in food as well as other materials which it needs from its external environment, and such processes we call them as endocytosis. ऐसे processes को हम क्या बोलेंगे? Endocytosis process. And amoeba is one such organism which acquires its food through this endocytosis process. Okay, so because this plasma membrane, which we also call it as cell membrane, okay, which is also called as cell membrane, वो क्या करते हैं, अपना खाना कैसे लेते हैं? They are going to engulf whatever food they come in contact with, or whatever material they wanted, they are going to engulf that from its external environment. And ऐसे process को हम क्या बोलेंगे? Endocytosis. So I want you to write this last statement which has been given, that the flexibility of the cell membrane enables the cell to engulf the food and other material from its external environment through a process known as endocytosis. And amoeba ka example you can write that amoeba is one such example that is acquiring its food through the process of endocytosis. Okay, so please take a note of this last sentence which has been given. Okay, so after that we have the another component to discuss, the most important component of the cell that we are having, which is the cell wall. So first component हमने discuss कर लिया, that is the plasma membrane. The second component that we have is the cell wall. Can you see the cell wall? So अभी I told you that the cell membrane, it act as a paint that we give it on the wall, and cell wall is the exact wall that we are having. तो प्लाज्मा मेम्ब्रेन क्या करता है? एज आई टोल्ड यू दैट इज़ अ पेंट, तो वो इंटरनल और एक्सटर्नल एनवायरनमेंट को सेपरेट करने में हेल्प करता है, राइट? नाउ लेट्स सी व्हाट डज द सेल वॉल डू. So plant cells, in addition to plasma membrane, they have another rigid outer covering. What is the cell wall? It is the rigid outer covering which is present in the cell, and we call this rigid outer covering as the cell wall. क्या बोलेंगे हम उसको? Cell wall. Okay. Now cell wall lies outside the plasma membrane. Cell wall कहाँ पे है? Plasma membrane के बाहर. Okay, Cell wall कहाँ पे? It lies outside the plasma membrane and the plant cell wall is mainly composed of cellulose. Plant cell wall किससे बनता है? Plant cell wall is composed of cellulose. Okay, so cellulose क्या है अभी? Cellulose is a complex substance which provides the structural strength to the plant, because I told you that this is a rigid outer covering. Rigid means hard. So cell wall एक hard outer covering है which is giving a structure to the plant. Why the plant is getting that structure? Because the plant का cell wall is mainly composed of cellulose, and cellulose एक ऐसा complex substance है जो plants को structural strength provide करता है. Okay, so when a living plant cell loses water through osmosis, see अगर एक living plant cell है मेरे पास and that living plant cell is losing water. Now we all know that plant can be lost or it can be water can be gained, right? So here in this case, if the plant is losing water through osmosis, you will see that the plant cell will shrink. So there is a shrinkage or contraction of the content of the cell away from the cell wall. And ऐसे phenomenon में जहाँ पे plant cell is losing the water and there is shrinkage which is taking place, this phenomenon we call it as plasmolysis. ठीक है, लिसिस मतलब ब्रेक डाउन होना. सो यहाँ पे क्या हो रहा है? वाटर लूज हो रहा है. सो देर इज अ काइंड ऑफ ब्रेक डाउन विच इज हैपनिंग हियर, राइट? So I want you all to take a note of it. When a living plant cell loses water through osmosis, there is going to be a shrinkage or contraction of the content of the cell away from the cell wall, and this phenomenon we call it as plasmolysis. Okay, please write this definition for plasmolysis. Okay, so you should know what is plasmolysis, you should know what is diffusion, you should know what is hypotonic solution, isotonic solution, and hypotonic solution. Okay, so now we have another activity to do. See, mount the peel of Rio leaf in water. Now Rio is a plant. You have to take the Rio leaf in water and then you have to take it on slide and you have to examine the cell like we have examined it for the other type of onion peel, right? So आप उसको examine करोगे cell को under high power microscope, compound microscope. Okay, there you will see there are some small green granules which are present, and that small green granules को we call them as chloroplast. Okay, and ये chloroplast is a thing which contains a green substance. Chloroplast के अंदर क्या होता है? We have a green substance which is present inside this chloroplast, and we call that green substance as chlorophyll. Chlorophyll कहाँ पे present है? Chloroplast के अंदर. And we all know this one thing, that why plants can prepare their own food and why animals cannot. or we human beings cannot make their own food because we don't have this chloroplast which is present within us, and that is the only reason why plants can prepare their own food and why we human beings and animals we cannot. Okay, so what you need to do is you need to put a strong solution of sugar or salt. क्या करेंगे हम, एक strong solution लेंगे sugar का या salt का, and we are going to add that on the mounted leaf of the slide. आपने जो slide ready किया है अभी, you have to put that strong solution of sugar or salt on that slide. You need to wait for a minute and then you have to again observe that under a microscope. Now what you will see, since you have added the concentrated solution of salt and water, what were we seeing? You will see the same thing. Okay, now place some rio leaves in boiling water for few minutes. अब हम क्या करेंगे, rio rio leaves को boiling water में डालेंगे कुछ times के लिए, कुछ minutes के लिए. So when you put that into the boiling water, this is going to kill the cells. Okay, so then now mount one leaf on a slide and observe it under a microscope. Now again put a strong solution of sugar and salt on that leaf of the slide and wait for 1 minute and observe it again. What did you find? यहाँ पे plasmolysis हुआ है. You know what plasmolysis is, right? Plasmolysis में क्या हो रहा है? The plant cell is going to lose the water and you will see that there will be a shrinkage. So whenever you come in contact with, अगर हम concentrated solution डाल रहे हैं अगर, तो वहाँ पे सेल में श्रिंकेज ही होगा. वाई सो? बिकॉज वाटर लूज करेगा हमारा सेल. दैट इज द रीजन यहाँ पे प्लाज्मोलिसिस होगा या नहीं होगा. सी, what do we infer from this activity is, it appears that only living cell and not dead cells are able to absorb the water by osmosis. ठीक है, तो सिर्फ लिविंग सेल्स ही ऑब्जर्व कर सकते हैं वाटर को बाय द प्रोसेस कॉल्ड एज ऑस्मोसिस. इफ द सेल्स आर डाइड, बिकॉज यहाँ पे हमने बोला दैट द सेल्स आर डेड नाउ, we have killed the cell by boiling the rio leaves, right? So now the cells are killed and that is the reason they are not going to absorb this water by osmosis process. So cell wall is going to permit the cells of the plants, fungi and bacteria so that they can withstand this very dilute external media without bursting. हम क्या करेंगे, जो भी cell wall है plants का, fungi का, bacteria का, they can withstand this dilute external media without bursting themselves. So in such media, the cells tend to take up the water. यहाँ पे ऐसे मीडियम में क्या होगा, the cells are going to take up the pressure against the cell, like take up the water by the process of osmosis. तो यहाँ पे cells ने water लिया. If the cells are taking water, what is going to happen? They're going to swell up, right? So the cells, they swell and this is going to build up pressure against the cell wall. So now, because the cell wall is getting a pressure, the wall is also going to exert an equal pressure against the swollen cell. And because of their walls, such cells, they can withstand much greater changes which can be seen in the surrounding medium than the animal cell. So this is the only reason how these cells are withstanding these greater changes which are taking place in the surrounding medium. Okay, now see, this is how you have to take. This is the lily leaf. आप उसको fold करोगे and you have to pull them apart. And this is the leaf peel that you get out of it. अब आप क्या करोगे? This is the petri dish that we are having. आप उसके अंदर water डालोगे. क्या करोगे आप? पेट्री डिश के अंदर वाटर डालोगे and you're going to add the leaf peel के pieces. Now you have to take one leaf piece. क्या करेंगे हम? एक लीफ पीस लेंगे स्लाइड के ऊपर, उसके ऊपर कवर स्लिप डालेंगे and then we are going to observe that under the microscope. And this is how you're going to see. These are the leaf cells which are present. We have guard cells which are present, as well as we have stomata which is being present. Okay. Okay. Then the next part that we have is nucleus. So let's discuss on nucleus as well, or should we discuss that in the next part? Okay, let's discuss this nucleus as well. So this is how the nucleus looks like. And if you remember in the past first part when we were discussing about the prokaryotic and the eukaryotic cell, there we saw that the prokaryotes, they don't have a well developed nuclear envelope, whereas the eukaryotes, they have this well developed nuclear envelope, right? So in a cell which is not dividing, now we all know cell division hota hai, right? So if we are talking about a cell which is not dividing, उसके डीएनए के अंदर देयर इज अ पार्ट ऑफ क्रोमैटिन मटेरियल विच इज प्रेजेंट, एंड वी ऑल नो दैट डीएनए के अंदर क्या होता है, क्रोमोसोम. क्रोमैटिन मटेरियल मींस क्रोमोसोम, सो डीएनए हैव अ क्रोमोसोम विच इज प्रेजेंट, राइट? सो दिस क्रोमैटिन मटेरियल इज विजिबल एज एन एंटैकल्ड मास ऑफ थ्रेड लाइक स्ट्रक्चर. तो हमारा क्रोमैटिन कैसा दिखता है पता है आपको? द क्रोमैटिन इज लाइक दिस थ्रेड लाइक स्ट्रक्चर्स आर कॉल्ड एज क्रोमैटिन. तो ऐसा क्रोमैटिन हमारे डीएनए के अंदर प्रेजेंट होता है. ओके, सो दिस क्रोमैटिन, इट लुक्स लाइक अ thread like structure. Okay, so whenever the cell is about to divide, abhi there are two types of cell division that we are going to discuss on, mitosis and meiosis. तब आपको समझेगा कि कैसे सेल डिवाइड कर रहा है, राइट? बट बिफोर दैट नाउ रिमेंबर कि जब भी सेल डिवाइड करता है, ये क्रोमैटिन मटेरियल जो है वो खुद को ऑर्गेनाइज करेगा एंड इट इज गोइंग टू गेट ऑर्गेनाइज्ड इनटू अ क्रोमोसोम. ओके, सो इट इज़ गोइंग टू फॉर्म अ क्रोमोसोम लाइक दिस. ऐसा करके वो क्रोमैटिन फाइबर से हमें क्या मिलता है, क्रोमोसोम. दिस इज द क्रोमैटिन फाइबर एंड दिस इज द क्रोमोसोम. एंड व्हेन दिस थिंग्स हैपन, क्रोमैटिन फाइबर क्रोमोसोम में कब ऑर्गेनाइज होगा? जब सेल डिवाइड करने वाला होगा तब. Okay, so nuclei, these are the specialized part of certain chromosome, because these chromosomes, they carry multiple copies of DNA, and this multiple copies of DNA, they are used to synthesize the ribosomal DNA. ठीक है, ये जो multiple copies है उसको हम क्यों use करते हैं? So that we can synthesize a ribosomal DNA there. Okay, so I hope this much is clear. क्रोमैटिन फाइबर जो है, क्रोमैटिन मटेरियल, हाउ इट इज developing itself, how it is organizing itself into a chromosome. Okay, so see, now the nucleus has a double layered covering, and that double layer covering of the nucleus, we call it as a nuclear membrane. क्या बोलेंगे हम उसको? nuclear membrane. And if you can see, the nuclear membrane here have some nuclear pores which are present. Can you see? These are the nuclear pores which have been present in the nuclear membrane, right? So see, they have a double layered covering called as nuclear membrane, and the nuclear membrane has pores which allow the transfer of material from inside the nucleus to the outside, that is to the cytoplasm, which we are going to discuss later. हम cytoplasm पे भी discuss करेंगे, but for now remember कि nucleus के ऊपर nuclear pore present होता है जो allow करता है in exchanging the material inside the cell as well as outside the cell. Okay, so now this nucleus, it contains this chromosome, and these chromosomes, they are visible as a rod shaped structure only when the cell is about to divide. As I told you that chromatin thread जैसा होता है, and when it is going to divide, it is going to form a rod shaped structure. Okay, so chromosome के पास क्या होता है, हमारा जो भी inherited character है, जो भी हमारे पास parents से जो भी information pass on होता है generation to generation, that is being passed to this chromosome, right? So chromosome contains some information for inheritance of character from parents to the next generation in the form of DNA. DNA stands for deoxyribonucleic acid molecule, right? तो अभी हमारा क्रोमोसोम किससे बना हुआ है? हमारा क्रोमोसोम बनता है डीएनए एंड प्रोटीन से, एंड डीएनए मॉलिक्यूल जो है उसके पास सारा इंपॉर्टेंट इन्फॉर्मेशन होता है विच इज़ नेसेसरी फॉर कंस्ट्रक्टिंग द सेल एज वेल एज फॉर ऑर्गेनाइजिंग द सेल. तो सेल के कंस्ट्रक्शन में एंड सेल को ऑर्गेनाइज करने में हमें किसकी ज़रूरत है? डीएनए मॉलिक्यूल की ज़रूरत है. एंड द फंक्शनल सेगमेंट्स ऑफ डीएनए दैट वी आर हैविंग, वी कॉल दैट फंक्शनल सेगमेंट एज जीन. तो अगर एक ऐसा सेल है जो डिवाइड नहीं कर रहा, वहाँ पे डीएनए इज गोइंग टू बी प्रेजेंट एज अ पार्ट ऑफ क्रोमैटिन मटेरियल. डीएनए कैसे प्रेजेंट होगा वहाँ पे? इट इज़ गोइंग टू बी प्रेजेंट एज अ क्रोमैटिन मटेरियल. ओके, सो दिस क्रोमैटिन मटेरियल एज आई टोल्ड यू क्रोमैटिन कैसे विजिबल होता है, इट इज़ विजिबल एज अ थ्रेड लाइक स्ट्रक्चर, एंड जैसे ही सेल डिवाइड करेगा इट इज़ गोइंग टू बिकम अ रॉड लाइक स्ट्रक्चर, एग शेप्ड, राइट. सो क्रोमैटिन जो है वो ऑर्गेनाइज हो जाएगा क्रोमोसोम में, एंड योर द न्यूक्लियस प्लेज अ सेंट्रल रोल. किसका रोल है यहाँ पे सबसे ज़्यादा? न्यूक्लियस इज गोइंग टू प्ले अ सेंट्रल रोल योर, बिकॉज the process by which a single cell divides and it forms two new cell, that is called as cellular reproduction. So nucleus is going to play a very important role here in cellular respiration, and it also plays a crucial part along with the environment, because it is going to help in determining the way how the cells are developing and what form it is going to exhibit when it matures, because there are certain chemical activities which takes place of the cell, right? So this nucleus plays a very crucial role part here as well. So when it comes to discussing about some organisms like bacteria ho gaye, then we have nuclear region of the cell. These are all poorly defined. Why? Because ये बैक्टीरिया और न्यूक्लियर रीजन होते हैं सेल के, उनके पास न्यूक्लियर मेम्ब्रेन नहीं होता. So because of the absence of this nuclear membrane, they are poorly defined. Okay, so such an undefined nuclear region which is having only this nucleic acid, we call that region as poorly defined nuclear region only with this nucleic acid, we call that as nucleoid. एंड सच ऑर्गेनिज्म जिनके पास न्यूक्लियर मेम्ब्रेन नहीं होता उनको हम बोलते हैं प्रोकैरियोट्स. क्या बोलते हैं? प्रोकैरियोट्स. एंड जिनके पास न्यूक्लियर मेम्ब्रेन होता है उनको हम बोलते हैं यूकैरियोट्स. नाउ प्रोकैरियोट्स का मीनिंग ही क्या है? प्रो मीन्स प्रिमिटिव और प्राइमरी, एंड कैरियोट्स मीन्स न्यूक्लियस. So primitive nucleus or primary nucleus is prokaryote. So who is having a well developed nuclear membrane? Those organisms are eukaryotes, and those who are not having a well developed nuclear membrane, they are called as prokaryotes. So this is one differentiation between prokaryotes and eukaryotes. Okay, now we are going to continue the rest of the part in the next class, where we are going to discuss on the prokaryotic cell and the eukaryotic cell. After that we are going to discuss on cytoplasm in detail. तो साइटोप्लाज्म जो है इट इज़ अ फ्लूइड लाइक स्ट्रक्चर, एंड सिंस दिस साइटोप्लाज्म इज समवॉट स्टिकी, जो भी सेल के ऑर्गेनल्स हैं ना वो स्टिक हो जाते हैं साइटोप्लाज्म पे. विल डिस्कस दैट इन द नेक्स्ट क्लास. देन वी विल बी डिस्कसिंग ऑन डिफरेंट सेल ऑर्गेनल्स which are present. So we have endoplasmic reticulum, which is one of the cell organelle that we are having. After that we have this Golgi apparatus. Then we have lysosomes to discuss. Then mitochondria to discuss, which is known as the powerhouse of the cell. After mitochondria we have plastids to discuss. Now this plastids, they are only present in the plant cell and they are absent in the animal cell. तो animal cells के पास plastid नहीं होता. Plastid is present only in the plant cell. After that we have this vacuoles to discuss, and then we will be discussing about the cell division in short. That is, we will be discussing about mitosis and meiosis. Then this is the summary of what we have already discussed in this lesson. We'll be discussing that. Okay, in short, फिर हम plant cell and animal cells पे differentiation देखेंगे. We will be discussing the differentiating point between prokaryotic cell and eukaryotic cell. आफ्टर दैट वी विल डिस्कस ऑन कि अगर प्लाज्मा मेम्ब्रेन रप्चर करता है या ब्रेक डाउन करता है, सो व्हाट विल हैपन इफ इट इज़ रप्चरिंग. ओके, देर अगर गोल्गी अपैरेटस नहीं होता तो व्हाट विल हैपन टू द लाइफ ऑफ सेल अगर इफ देर इज़ नो गोल्गी अपैरेटस. विल डिस्कस दैट. देन वी हैव अनदर क्वेश्चन वेयर विल सी कि which organelle is known as the powerhouse of the cell, and why do we call that organelle as the powerhouse of the cell. Okay, after that we have the question as where do the lipids and proteins they constitute the cell membrane and where did they get synthesized. तो lipids and proteins synthesized कहाँ पे होते हैं and where like this lipids and proteins are constituting the cell membrane. After that we have this question as how does an amoeba obtain its food. Okay, so we have discussed this today and I hope you know the answer for this कि amoeba अपना food कैसे obtain करता है. Then what is osmosis also we have discussed, right? Then this is an experiment or an activity which we will be discussing. And lastly we have this question as which type of cell division is required for the growth and repair of body and which type is involved in the formation of gamete. So the answer to all these 10 questions are very much easy once you understand the whole lesson or once we just complete with the lesson. Once we complete with the whole lesson, you will get an idea. You will get an idea on this fundamental unit of life. Okay, so I hope you are clear with whatever we have discussed till now in this lesson, fundamental unit of life. The rest part of the lesson we are going to continue in the next part. Thank you everyone.